At the tissue level, oxygen is released from oxyhemoglobin and diffuses into cells for metabolic activities. Simultaneously, CO produced during cellular respiration diffuses into the blood and is transported back to the lungs for elimination. Waste elimination. 1. Role of the lungs in CO removal. CO is a metabolic waste product that must be eliminated to maintain blood pH and prevent acidosis. In mammals, CO is transported via blood and exhaled through the lungs. 2. Alternative gas waste elimination pathways. Aquatic organisms, fish, amphibians eliminate CO directly into water through gills. Insects eliminate CO through tracheal spiracles. Some reptiles and amphibians use cutaneous respiration to eliminate gases through the skin. Previous year question. Question. Describe the transport of respiratory gases in mammals and explain how oxygen and carbon dioxide are exchanged at the alveolar and tissue levels. Answer. In mammals, oxygen transport occurs via hemoglobin in red blood cells. Oxygen binds to hemoglobin in the lungs to form oxyhemoglobin, which travels through the bloodstream. At the tissue level, oxygen is released and diffuses into cells for metabolic use. Carbon dioxide, a metabolic waste product, is transported in three main forms. Bicarbonate ions, HCO, 60-70%, formed by the dissociation of carbonic acid in red blood cells. Carbaminohemoglobin, 20-30%, CO binds to hemoglobin. Dissolved CO, 7-10%, directly dissolved in plasma. Gas exchange occurs via diffusion based on partial pressure gradients. In the lungs, oxygen diffuses into blood, and CO diffuses out. At tissues, oxygen is released, and CO enters the blood for transport back to the lungs. Multiple choice questions, MCQs. 1. Which of the following factors enhances oxygen unloading from hemoglobin in tissues? A. High pH. B. Low CO concentration. C. High temperature. D. Low 2. 3 BPG levels. Answer. C. High temperature. 2. What is the primary mechanism of CO transport in the blood? A. Bound to hemoglobin is carbaminohemoglobin. B. Dissolved directly in plasma. C. Is bicarbonate ions. HCO. D. Through lymphatic circulation. Answer. C. Is bicarbonate ions. HCO. 3. In mammals, gas exchange occurs primarily in a. Trachea. B. Bronchi. C. Alveoli. D. Pleural cavity. Answer. C. Alveoli. This detailed lecture provides complete coverage of anatomical considerations, gas transport, gas exchange, and waste elimination for your University of Calicut PhD entrance exam in zoology. Let me know the next topic. You said, neural and chemical regulation of respiration. ChatGPT said, neural and chemical regulation of respiration. Introduction. Respiration is a highly regulated process controlled by both neural and chemical mechanisms to maintain homeostasis. The body must adjust breathing rates based on oxygen demand, carbon dioxide levels, and pH balance. The nervous system controls respiration through signals from the brain, while chemoreceptors detect changes in blood gases and pH, ensuring proper gas exchange and metabolic function. Neural regulation of respiration. Respiratory control is primarily governed by the brainstem, specifically the medulla oblongata and pons, which regulate the rhythmic pattern of breathing. 1. Medullary respiratory centers, rhythmicity centers. The medulla oblongata contains the primary respiratory control centers that regulate the basic rhythm of breathing. Dorsal respiratory group, DRG, controls inspiration by sending signals to the diaphragm and external intercostal muscles. Ventral respiratory group, VRG, controls forced expiration and modulates breathing during exercise. 2. Pontine respiratory centers, regulation of breathing pattern. The pons contains two centers that fine-tune the rhythm set by the medulla. Pneumataxic center, inhibits inspiration to prevent overinflation of the lungs. Apneustic center, stimulates prolonged inspiration, working in opposition to the pneumataxic center. 3. Higher brain centers, voluntary control of breathing. The cerebral cortex allows voluntary control of breathing, e.g., holding breath or deep breathing. The limbic system and hypothalamus can alter respiration based on emotions, e.g., anxiety or fear can increase breathing rate. Chemical regulation of respiration. The respiratory system responds to changes in blood oxygen, O, carbon dioxide, CO, and pH through specialized sensors called chemoreceptors. 1. Central chemoreceptors, brainstem regulation. Located in the medulla oblongata, these receptors primarily detect CO levels and pH changes in cerebrospinal fluid, CSF. High CO, hypercapnia, or low pH, acidosis, triggers an increase in respiratory rate to expel CO. Low CO, hypocapnia, or high pH, alkalosis, slows respiration. 2. Peripheral chemoreceptors, blood gas sensors. Located in the carotid bodies, at the bifurcation of the carotid artery, and aortic bodies near the aortic arch. These receptors are sensitive to low O levels, hypoxia, right pointing arrow increases respiration. High CO levels, hypercapnia, right pointing arrow increases respiration. Low pH, acidosis, right pointing arrow stimulates breathing to remove CO and restore pH balance. Integration of neural and chemical control. 1. Negative feedback mechanism. The respiratory system maintains gas homeostasis via a negative feedback loop. Example, if CO levels rise, chemoreceptors stimulate the respiratory center to increase breathing rate, eliminating excess CO and restoring balance. 2. Reflex control of respiration. Herring-Brewer reflex, prevents lung overinflation by inhibiting inspiration via stretch receptors in the lungs. Irritant receptors, located in airways, trigger coughing or sneezing in response to irritants. J receptors, juxtapulmonary capillary receptors, respond to pulmonary congestion and trigger rapid, shallow breathing. Previous year question. Question. Describe the role of chemoreceptors in the regulation of respiration and explain how CO levels influence respiratory rate. Answer. Chemoreceptors play a crucial role in regulating respiration by detecting changes in blood gas composition and pH. Central chemoreceptors, located in the medulla oblongata, primarily respond to CO levels and pH changes in cerebrospinal fluid, CSF. When CO levels rise, hypercapnia, it forms carbonic acid, lowering CSF pH. This stimulates the respiratory center to increase breathing rate, expelling excess CO and restoring pH balance. Peripheral chemoreceptors, found in the carotid and aortic bodies, detect low oxygen, hypoxia, high CO, and blood pH changes. When oxygen levels drop or CO levels rise, these receptors signal the brainstem to enhance ventilation, improving oxygen intake and CO removal. Thus, CO is the primary driver of respiration, with even small increases triggering rapid breathing, hyperventilation, while low CO levels cause slower respiration, hypoventilation. Multiple choice questions, MCQs. 1. The primary stimulus for breathing under normal conditions is low oxygen levels. B. High carbon dioxide levels. C. High pH levels. D. Low body temperature. Answer. B. High carbon dioxide levels. 2. Which part of the brain contains the primary respiratory centers? A. Cerebrum. B. Hypothalamus. C. Medulla oblongata. D. Thalamus. Answer. C. Medulla oblongata. 3. The Hering-Breuer-Lex prevents A. Hyperventilation. B. Overinflation of the lungs. C. Decrease in breathing rate. D. Excess CO retention. Answer. B. Overinflation of the lungs. This detailed lecture provides complete coverage of neural and chemical regulation of respiration for your University of Calicut PhD entrance exam in zoology. Let me know the next topic. You said. Nerve system and sense organs. Neurons. Action potentia. ChatGPT said. Nervous system and sense organs. Neurons and action potential. Introduction. The nervous system is a complex network that coordinates bodily functions, processes sensory information, and regulates responses. It consists of neurons, the fundamental units, and glial cells, supportive cells. The transmission of electrical signals occurs via action potentials, which allow communication between neurons and target tissues. This section explores the structure and function of neurons, mechanisms of action potentials, and their role in nervous system communication. Neurons. Structure. Function 1. Structure of a neuron. Neurons are specialized cells responsible for transmitting electrical and chemical signals. They have 3 main parts. Cell body. Soma contains the nucleus and essential organelles. It processes incoming signals. Dendrites. Short, branched extensions that receive signals from other neurons. Axon. A long, single projection that transmits impulses away from the cell body toward other neurons or effector organs. At the axon terminal, neurons communicate via synapses, where electrical signals are converted into chemical signals using neurotransmitters. 2. Types of neurons. Sensory neurons afferent transmit signals from sensory receptors to the central nervous system, CNS. Motor neurons afferent carry signals from the CNS to muscles and glands. Interneurons act as relay neurons within the CNS, connecting sensory and motor neurons. Action potential, the electrical signal of neurons. An action potential is a rapid electrical change across the neuronal membrane that enables nerve impulse transmission. It follows an all or none principle, meaning it either occurs fully or not at all. 1. Resting membrane potential, RMP. In the resting state, the neuron maintains a negative charge, minus 70 millivolts, inside the cell compared to the outside. This is maintained by the sodium-potassium, not K pump, which actively transports 3 Na ions out and 2 K ions in, creating an electrochemical gradient. Leaky K channels allow some potassium to diffuse out, further maintaining negativity inside. 2. Phases of an action potential. Depolarization. When a stimulus reaches the neuron, voltage-gated Na channels open, allowing Na to rush in. This causes the membrane potential to become positive, plus 30 millivolts. Repolarization. Na channels close, and voltage-gated K channels open, allowing K to leave the neuron, restoring negativity. Hyperpolarization. Excessive K leaves the neuron, making it more negative than the resting potential before returning to baseline. Refractory period. The neuron temporarily becomes unresponsive to further stimulation, ensuring unidirectional signal transmission. 3. Conduction of action potential. In unmyelinated neurons, the action potential propagates continuously along the axon. In myelinated neurons, action potentials jump between nodes of Ranvier via saltatory conduction, increasing signal speed. Role of action potential in neural communication. Action potentials allow neuronal signaling, muscle contractions, reflex actions, and brain functions such as learning and memory. Neurotransmitters like acetylcholine, dopamine, serotonin, and glutamate mediate synaptic communication. Previous year question. Question. Describe the phases of an action potential and explain its role in neuronal signaling. Answer. An action potential is a rapid electrical event in neurons that allows signal transmission. It has 4 key phases. Depolarization. Na enters the neuron, making the inside positive. Repolarization. K exits, restoring the negative charge. Hyperpolarization. Excess K leaves, making the neuron momentarily more negative than usual. Refractory period. The neuron resets and becomes ready for the next impulse. Action potentials enable neuronal communication, muscle contractions, and reflexes. In myelinated neurons, impulses travel faster due to saltatory conduction between nodes of Ranvier. Multiple choice questions. MCQs 1. What is the resting membrane potential of a neuron? A plus 30 millivolts. B minus 55 millivolts. C minus 70 millivolts. D 0 millivolts. Answer. C minus 70 millivolts. 2. During depolarization, which ion enters the neuron? A K. B CLC Na D Ca squared. Answer. C Na. 3. What is the function of the refractory period in neurons? A. Speeds up conduction. B prevents backward propagation of impulses. C increases synaptic activity. D inhibits neurotransmitter release. Answer. B prevents backward propagation of impulses. This detailed lecture on neurons and action potentials provides full coverage for your University of Calicut PhD entrance exam in zoology. Let me know the next topic. You said. Gross neuroanatomy of the brain and spinal cord. ChatGPT said. Gross neuroanatomy of the brain and spinal cord. Introduction. The central nervous system, CNS, consists of the brain and spinal cord, which control all voluntary and involuntary functions of the body. The brain is the command center, processing sensory information and coordinating responses, while the spinal cord serves as the primary communication pathway between the brain and the peripheral nervous system. Understanding the gross neuroanatomy of these structures is crucial for comprehending their functions and interconnections. Brain, structure and functions. The brain is a highly specialized organ encased within the skull, protected by meninges dura mater, arachnoid mater, and pia mater, and cushioned by cerebrospinal fluid, CSF. It consists of 4 main regions. 1. Cerebrum. The largest part of the brain, responsible for conscious thought, sensory perception, voluntary movement, and higher cognitive functions. It is divided into 2 hemispheres, left and right, connected by the corpus callosum. 4 lobes. Frontal lobe, controls reasoning, problem-solving, voluntary movements, and speech. Broca's area. Parietal lobe, processes touch, temperature, and pain. Somatosensory cortex. Temporal lobe, involved in auditory processing, language comprehension. Wernicke's area, and memory. Occipital lobe, processes visual information. 2. Diencephalon. Located beneath the cerebrum, it includes thalamus, relay center for sensory signals except smell. Hypothalamus, regulates homeostasis, hunger, thirst, temperature, and hormone secretion via the pituitary gland. Epithalamus pineal gland controls circadian rhythms by secreting melatonin. 3. Brainstem. The brainstem connects the brain to the spinal cord and controls autonomic functions such as breathing and heartbeat. It consists of midbrain, controls reflexive eye and auditory movements. Pons, relays signals between the cerebrum and cerebellum, regulates sleep and respiration. Medulla oblongata, controls involuntary functions such as heartbeat, breathing, and blood pressure. 4. Cerebellum. Located at the back of the brain, it coordinates movement, balance, and posture. It has two hemispheres and a central region called the vermis. Spinal cord, structure and functions. The spinal cord is a cylindrical structure extending from the medulla oblongata to the lower back L1-L2 level. It is encased in vertebrae and protected by CSF. 1. Structure of the spinal cord. Divided into cervical, thoracic, lumbar, sacral, and coccygeal regions. Contains gray matter inner region, neuron cell bodies, and white matter outer region, myelinated axons. Central canal, contains cerebrospinal fluid. 2. Spinal cord functions. Acts as the main communication highway between the brain and peripheral nervous system. Contains reflex arcs, enabling automatic responses without involving the brain. Has ascending tracts sensory pathways to the brain and descending tracts motor pathways from the brain to muscles. Previous year question. Question. Describe the gross anatomy of the human brain and its functional divisions. Answer. The human brain is divided into 4 main regions. Cerebrum, the largest part, responsible for cognition, voluntary movement, and sensory perception. It is divided into frontal, parietal, temporal, and occipital lobes. Diencephalon, includes the thalamus, sensory relay, hypothalamus, homeostasis and endocrine regulation, and pineal gland circadian rhythms. Brainstem, consists of the midbrain, pons, and medulla oblongata, which control autonomic functions such as respiration and heart rate. Cerebellum, coordinates movement and balance. The spinal cord extends from T. Gross neuroanatomy of the brain and spinal cord introduction. The central nervous system, CNS, consists of the brain and spinal cord, which control all voluntary and involuntary functions of the body. The brain is the command center, processing sensory information and coordinating responses, while the spinal cord serves as the primary communication pathway between the brain and the peripheral nervous system. Understanding the gross neuroanatomy of these structures is crucial for comprehending their functions and interconnections. Brain, structure and functions. The brain is a highly specialized organ encased within the skull, protected by meninges, dura mater, arachnoid mater, and pia mater, and cushioned by cerebrospinal fluid, CSF. It consists of 4 main regions. 1. Cerebrum. The largest part of the brain, responsible for conscious thought, sensory perception, voluntary movement, and higher cognitive functions. It is divided into 2 hemispheres, left and right, connected by the corpus callosum. 4 lobes. Frontal lobe, controls reasoning, problem-solving, voluntary movements, and speech-Broca's area. Parietal lobe, processes touch, temperature, and pain, somatosensory cortex. Temporal lobe, involved in auditory processing, language comprehension, Wernicke's area, and memory. Occipital lobe, processes visual information. 2. Diencephalon. Located beneath the cerebrum, it includes thalamus, relay center for sensory signals except smell. Hypothalamus, regulates homeostasis, hunger, thirst, temperature, and hormone secretion via the pituitary gland. Epithalamus, pineal gland, controls circadian rhythms by secreting melatonin. 3. Brainstem. The brainstem connects the brain to the spinal cord and controls autonomic functions such as breathing and heartbeat. It consists of midbrain, controls reflexive eye and auditory movements. Pons, relays signals between the cerebrum and cerebellum, regulates sleep and respiration. Medulla oblongata, controls involuntary functions such as heartbeat, breathing, and blood pressure. 4. Cerebellum. Located at the back of the brain, it coordinates movement, balance, and posture. It has 2 hemispheres and a central region called the vermis. Spinal cord, structure and functions. The spinal cord is a cylindrical structure extending from the medulla oblongata to the lower back L1-L2 level. It is encased in vertebrae and protected by CSF. 1. Structure of the spinal cord. Divided into cervical, thoracic, lumbar, sacral, and coccygeal regions. Contains gray matter inner region, neuron cell bodies, and white matter outer region, myelinated axons. Central canal, contains cerebrospinal fluid. 2. Spinal cord functions. Acts as the main communication highway between the brain and peripheral nervous system. Contains reflex arcs, enabling automatic responses without involving the brain. Has ascending tracts sensory pathways to the brain and descending tracts motor pathways from the brain to muscles. Previous year question. Question. Describe the gross anatomy of the human brain and its functional divisions. Answer. The human brain is divided into 4 main regions. Cerebrum, the largest part, responsible for cognition, voluntary movement, and sensory perception. It is divided into frontal, parietal, temporal, and occipital lobes. Diencephalon, includes the thalamus, sensory relay, hypothalamus, homeostasis and endocrine regulation, and pineal gland circadian rhythms. Brainstem, consists of the midbrain, pons, and medulla oblongata, which control autonomic functions such as respiration and heart rate. Cerebellum, coordinates movement and balance. The spinal cord extends from the brainstem, transmitting sensory and motor signals between the brain and body. It is divided into cervical, thoracic, lumbar, sacral, and coccygeal regions, with gray and white matter forming the core structure. Multiple choice questions, MCQs. 1. Which part of the brain is responsible for coordinating balance and movement? A. Thalamus. B. Cerebellum. C. Hypothalamus. D. Medulla oblongata. Answer. B. Cerebellum. 2. Corpus callosum connects. A. The cerebellum and spinal cord. B. The brainstem and thalamus. C. The 2 cerebral hemispheres. D. The pons and medulla. Answer. C. The 2 cerebral hemispheres. 3. The primary function of the spinal cord is to process emotions. B. Relay signals between the brain and body. C. Control circadian rhythms. D. Store long-term memory. Answer. B. Relay signals between the brain and body. This comprehensive lecture provides full coverage of the gross anatomy of the brain and spinal cord for your University of Calicut PhD entrance exam in zoology. Let me know the next topic. You said. Central and peripheral nervous system. ChatGPT said. Central and peripheral nervous system introduction. The nervous system is the primary control and communication system of the body. It is broadly classified into central nervous system, CNS, comprises the brain and spinal cord, responsible for processing information and coordinating responses. Peripheral nervous system, PNS, consists of nerves and ganglia outside the CNS, transmitting signals between the CNS and the rest of the body. Both systems work together to regulate voluntary and involuntary functions, including sensory perception, movement, homeostasis, and reflex actions. Central nervous system, CNS. 1. Structure of the CNS. The CNS is composed of Brain, the control center of the body, divided into the cerebrum, cerebellum, diencephalon, and brainstem. It processes sensory input, controls movement, and regulates higher cognitive functions. Spinal cord, a long cylindrical structure extending from the brainstem to the lower back, serving as a communication highway between the brain and peripheral nerves. 2. Functions of the CNS. Integrates sensory information and responds appropriately. Controls motor functions, including voluntary and involuntary movements. Regulates homeostasis, including heart rate, respiration, digestion, and body temperature. Facilitates higher order functions, such as memory, learning, emotions, and problem solving. Peripheral nervous system, PNS. The PNS connects the CNS to the body's limbs and organs, enabling communication between the brain, spinal cord, and external, internal environments. It is divided into two main components. 1. Somatic nervous system, SNS. Controls voluntary movements by sending motor signals to skeletal muscles. Carries sensory information from external stimuli, touch, pain, temperature, to the CNS via afferent neurons. Includes cranial nerves, 12 pairs, and spinal nerves, 31 pairs, that mediate sensory and motor functions. 2. Autonomic nervous system, ANS. Regulates involuntary functions such as heart rate, digestion, respiration, and glandular activity. Divided into sympathetic nervous system, fight or flight, response, increasing heart rate, dilating pupils, and redirecting blood flow to muscles. Parasympathetic nervous system, rest and digest, response, promoting relaxation, digestion, and energy conservation. Differences between CNS and PNS. The CNS processes and integrates information, while the PNS transmits signals between the CNS and the body. The CNS is protected by the skull and vertebrae, whereas the PNS consists of exposed nerves. The CNS controls higher cognitive functions, whereas the PNS mediates sensory and motor responses. Previous year question. Question. Differentiate between the central and peripheral nervous system, highlighting their roles and components. Answer. The central nervous system, CNS, includes the brain and spinal cord, responsible for processing, integrating, and responding to sensory and motor information. The peripheral nervous system, PNS, consists of nerves and ganglia that relay signals between the CNS and the body. The CNS has two main parts. Brain, controls voluntary and involuntary functions, emotions, and cognitive processes. Spinal cord, facilitates communication between the brain and peripheral nerves. The PNS is divided into somatic nervous system, SNS, controls voluntary muscle movements and transmits sensory information. Autonomic nervous system, ANS, regulates involuntary functions, with sympathetic and parasympathetic divisions balancing body responses. The CNS is the processing center, while the PNS acts as the communication network, ensuring body functions are coordinated efficiently. Multiple choice questions, MCQs. 1. Which part of the nervous system is responsible for voluntary muscle movements? Autonomic nervous system. B. Somatic nervous system. C. Sympathetic nervous system. D. Parasympathetic nervous system. Answer. B. Somatic nervous system. 2. The parasympathetic nervous system primarily functions to increase heart rate and blood pressure. B. Prepare the body for stress and emergencies. C. Conserve energy and promote digestion. D. Stimulate skeletal muscle contraction. Answer. C. Conserve energy and promote digestion. 3. The spinal cord is part of which nervous system? Peripheral nervous system. B. Somatic nervous system. C. Central nervous system. D. Autonomic nervous system. Answer. C. Central nervous system. This comprehensive lecture provides full coverage of the central and peripheral nervous system for your University of Calicut PhD entrance exam in zoology. Let me know the next topic. You said neural control of muscle tone and posture. ChatGPT said neural control of muscle tone and posture. Introduction. Muscle tone and posture are essential for maintaining stability, movement coordination, and balance. The nervous system plays a crucial role in regulating muscle tone, the continuous partial contraction of muscles, and posture, the alignment of body segments against gravity. Neural control of these functions involves sensory inputs, motor outputs, reflex circuits, and higher brain centers that coordinate movements and maintain equilibrium. Muscle tone, definition and regulation. 1. What is muscle tone? Muscle tone refers to the continuous and passive partial contraction of muscles, even when they are at rest. It ensures that muscles are ready for action and helps maintain body posture. 2. Neural mechanisms controlling muscle tone. Muscle tone is regulated by a reflex arcs and spinal cord circuits. The muscle spindle is a specialized sensory receptor within muscles that detects changes in muscle length and sends signals to the spinal cord. When a muscle stretches, the stretch reflex, myotatic reflex, is activated, causing the muscle to contract to maintain tone. Gamma motor neurons adjust the sensitivity of muscle spindles to maintain proper tension. The Golgi tendon organ, GTO, prevents excessive contraction by inhibiting motor neurons when muscle tension is too high. B. Brainstem and reticulospinal tract. The brainstem, especially the reticular formation, regulates background muscle tone through the reticulospinal tract. The vestibulospinal tract plays a role in adjusting muscle tone in response to changes in body position and balance. See cerebellum and basal ganglia. The cerebellum fine-tunes movements, ensuring smooth coordination and preventing excessive muscle stiffness or flaccidity. The basal ganglia modulate motor control by suppressing unwanted movements and adjusting muscle tone through the extrapyramidal system. D. Cortical control, motor cortex and descending pathways. The primary motor cortex, in the frontal lobe, initiates voluntary movements and fine-tunes muscle activation. Descending pathways, corticospinal and rubrospinal tracts, send signals to motor neurons to regulate muscle tone and movement precision. Posture, neural control and maintenance. 1. Definition of posture. Posture refers to the alignment and positioning of the body to maintain stability and balance against gravity. It is dynamically regulated by neural feedback mechanisms and muscle tone adjustments. 2. Neural control of posture. Sensory inputs for postural regulation. Vestibular system, located in the inner ear, it detects head position and motion, sending signals to maintain balance. Proprioceptors, found in muscles, tendons, and joints, they provide continuous feedback about body position. Visual system, helps in maintaining orientation by processing spatial information. B. Motor pathways controlling posture. Vestibulospinal tract, controls muscle tone in response to changes in head position, adjusting posture accordingly. Reticulospinal tract, regulates posture by modulating reflexes and muscle tone. Corticospinal tract, provides voluntary control of postural muscles. See role of the cerebellum and basal ganglia. The cerebellum integrates sensory inputs and fine-tunes postural adjustments. Damage to the cerebellum can cause ataxia, uncoordinated movements, and poor balance. The basal ganglia help maintain postural stability and regulate smooth transitions between movements. Disorders of muscle tone and posture. 1. Hypotonia, low muscle tone. Caused by damage to motor neurons, cerebellum, or spinal cord. Leads to flaccid paralysis and poor posture control. Seen in conditions like spinal cord injury and cerebral palsy. 2. Hypertonia, increased muscle tone due to dysfunction of the corticospinal or extrapyramidal systems. Results in rigidity, spasticity, and poor coordination. Seen in Parkinson's disease and upper motor neuron lesions. 3. Postural instability. Occurs in disorders like Parkinson's disease, where basal ganglia dysfunction affects postural reflexes. Cerebellar damage causes ataxia, affecting postural control. Previous year question. Question. Describe the neural mechanisms that control muscle tone and posture. How do different brain regions contribute to postural stability? Answer. Muscle tone and posture are regulated by multiple neural circuits involving the spinal cord, brainstem, cerebellum, basal ganglia, and cerebral cortex. Muscle tone, maintained by the muscle spindle reflex, involving sensory feedback from muscle stretch receptors. Gamma motor neurons adjust spindle sensitivity, while the reticulospinal and vestibulospinal tracts regulate background muscle tension. Postural control, the vestibular system, proprioceptors, and visual inputs provide sensory feedback for posture adjustment. The cerebellum fine-tunes movements, and the basal ganglia stabilize postural transitions. The motor cortex and descending pathways execute voluntary posture adjustments. Dysfunctions in these neural circuits lead to hypotonia, hypertonia, and postural instability, commonly seen in neurological disorders like Parkinson's disease, cerebral palsy, and cerebellar ataxia. Multiple choice questions MCQs. 1. Which neural structure is primarily responsible for fine-tuning muscle tone and coordination? A. Cerebellum. B. Thalamus. C. Hypothalamus. D. Medulla oblongata. Answer. A. Cerebellum. 2. The vestibulospinal tract plays a crucial role in a. Regulating voluntary movements. B. Controlling autonomic function. C. Adjusting posture and balance. C. Coordinating eye movements. Answer. C. Adjusting posture and balance. 3. Damage to the basal ganglia results in a. Loss of sensory function. B. Impaired coordination and muscle rigidity. C. Loss of autonomic control. D. Decreased hormone secretion. Answer. B. Impaired coordination and muscle rigidity. This detailed lecture provides full coverage of the neural control of muscle tone and posture for your University of Calicut PhD entrance exam in zoology. Let me know the next topic. You said. Vision, hearing and tactile response. ChatGPT said. Vision, hearing, and tactile response introduction. Sensory perception is fundamental for survival and adaptation. The visual, auditory, and tactile systems allow organisms to perceive their environment, process stimuli, and respond appropriately. These systems rely on specialized sensory receptors, neural pathways, and brain processing centers to convert external stimuli into meaningful sensory experiences. Vision. 1. Structure of the eye. The human eye is a highly specialized organ for detecting light and forming images. Its key components include cornea, transparent front layer that refracts light. Lens, adjusts shape for focusing, accommodation. Retina, contains photoreceptors, rods and cones that convert light into neural signals. Optic nerve, transmits visual information to the brain. Iris and pupil, regulate the amount of light entering the eye. 2. Photoreception and neural processing rods, highly sensitive to low light, night vision but lack color perception. Cones, detect color and fine detail, functioning best in bright light. Visual pathway, the optic nerve, right pointing arrow, optic chiasma, right pointing arrow, lateral geniculate nucleus, LGN, right pointing arrow, visual cortex in the occipital lobe. 3. Color vision and visual adaptation. Trichromatic theory, the eye detects 3 primary colors, red, green, blue, via cone cells. Opponent process theory, visual perception is based on opposing color pairs, red, green, blue, yellow. Dark and light adaptation, rods adapt to darkness slowly, while cones adjust quickly to bright light. 4. Disorders of vision. Myopia, nearsightedness, light focuses in front of the retina. Hyperopia, farsightedness, light focuses behind the retina. Color blindness, deficiency in cone cells, usually affecting red-green perception. Hearing, auditory system. 1. Structure of the ear. The ear is divided into outer ear, includes the pinna and auditory canal, which collects sound waves. Middle ear, contains the ossicles malleus, incus, stapes that amplify sound vibrations. Inner ear, houses the cochlea, where sound waves are converted into electrical signals. 2. Mechanism of hearing. Sound waves travel through the auditory canal and vibrate the tympanic membrane eardrum. Vibrations pass through the ossicles, amplifying the signal. The cochlea converts mechanical vibrations into electrical signals via hair cells in the basilar membrane. The auditory nerve transmits impulses to the auditory cortex in the temporal lobe for sound interpretation. 3. Frequency and pitch perception. Place theory, different frequencies activate specific locations on the basilar membrane. Frequency theory, neurons fire at the same frequency as the sound wave. Tonotopic organization, the cochlea and auditory cortex map sounds by frequency. 4. Hearing disorders. Conductive hearing loss, caused by blockage or damage to the middle ear. Sensory neural hearing loss, damage to the cochlear auditory nerve, e.g., due to aging or loud noise exposure. Tinnitus, perception of ringing sounds in the absence of external stimuli. Tactile response, somatosensory system. 1. Types of tactile receptors. Mechanoreceptors, detect touch, pressure, vibration. Merkel discs, light touch. Meissner's corpuscles, fine touch, low frequency vibrations. Pacinian corpuscles, deep pressure, high frequency vibrations. Ruffini endings, skin stretch and sustained pressure. Thermoreceptors, detect temperature changes. Nociceptors, respond to pain and harmful stimuli. 2. Neural pathway for touch perception. Tactile signals follow the dorsal column medial lemniscus DCML pathway. First order neurons carry sensory signals from the skin to the spinal cord. Second order neurons transmit signals to the thalamus. Third order neurons relay signals to the somatosensory cortex in the parietal lobe. 3. Tactile sensitivity and adaptation. 2-point discrimination, ability to distinguish too close stimuli, highest in fingertips. Sensory adaptation, receptors reduce response to constant stimuli, e.g., getting used to clothing. 4. Disorders of the somatosensory system. Neuropathy, damage to peripheral nerves, causing numbness or pain. Phantom limb syndrome, sensation in an amputated limb due to neuroplasticity. Previous year question. Question, describe the structure and function of the human auditory system. How does the cochlea convert sound waves into neural signals? Answer. The auditory system detects and processes sound through the outer ear, middle ear, and inner ear. Outer ear, the pinna collects sound waves and directs them through the auditory canal to the eardrum. Middle ear, the ossicles malleus, incus, stapes amplify vibrations and transmit them to the cochlea via the oval window. Inner ear, the cochlea, filled with fluid, contains the basilar membrane and hair cells that detect sound frequencies. Mechanism of sound transduction in the cochlea. Vibrations in the basilar membrane bend the stereocilia on hair cells. This bending opens ion channels, leading to depolarization and the release of neurotransmitters. The auditory nerve sends impulses to the brainstem, thalamus, and auditory cortex, where sound is processed. The cochlea's tonotopic organization ensures that high-frequency sounds are detected at the base, while low-frequency sounds are detected at the apex. Multiple choice questions MCQs. 1. The primary photoreceptors responsible for color vision in humans are rods. Beat cones. C. Bipolar cells. D. Ganglion cells. Answer: beat cones. 2. Which part of the ear is responsible for converting sound waves into neural signals? Ossicles. B. Membrane. C. Cochlea. D. Auditory canal. Answer: C. Cochlea. 3. Which sensory receptor is most sensitive to deep pressure and high-frequency vibration? Merkel discs. Meissner's corpuscles. C. Pacinian corpuscles. D. Ruffini endings. Answer: C. Pacinian corpuscles. This comprehensive lecture covers vision, hearing, and tactile response with maximum detail for your University of Calicut PhD entrance exam in zoology. Let me know the next topic. You said. Excretory system, comparative physiology of excretion, ChatGPT said. Excretory system, comparative physiology of excretion introduction. Excretion is a fundamental physiological process that eliminates metabolic wastes, toxins, and excess substances to maintain homeostasis. Different organisms have evolved diverse excretory mechanisms based on their habitat, metabolic processes, and evolutionary lineage. The primary waste products include ammonia, urea, and uric acid, which vary in toxicity and solubility, influencing the mode of excretion. 1. Modes of excretion. Organisms excrete nitrogenous waste in 3 major forms. 1.1 Ammonotalism. Ammonia excretion. Ammonia is the primary nitrogenous waste in ammonotelic animals. It is highly toxic and requires large amounts of water for excretion. Found in aquatic invertebrates, bony fishes, amphibians, and amphibian larvae, tadpoles, where ample water availability facilitates rapid diffusion of ammonia through gills or body surfaces. 1.2 Ureotalism. Urea excretion. Ureotelic organisms convert ammonia into the less toxic and water-soluble urea. This process occurs in the liver via the ornithine urea cycle, Krebs-Henselate cycle. Urea is then transported via the bloodstream to the kidneys for excretion in urine. Found in amphibians, frogs, toads, mammals, humans, dogs, whales, and cartilaginous fishes, sharks, rays. Ureotalism allows conservation of water, making it adaptively beneficial for terrestrial life. 1.3 Uricotalism. Uric acid excretion. Uricotelic animals excrete nitrogenous waste as uric acid, which is insoluble in water and excreted as a semi-solid paste. Uric acid is non-toxic and allows maximum water conservation and adaptation for arid environments. Found in birds, reptiles, snakes, lizards, insects, grasshoppers, cockroaches. 1.4 Other modes of excretion. Guanidalism. Some marine organisms, e.g., spiders and scorpions, excrete guanine instead of urea or uric acid. Aminodalism. Some echinoderms excrete free amino acids directly. 2. Comparative excretory organs in animals. 2.1 Protozoa and simple invertebrates. Diffusion and contractile vacuole. Protozoans, amoeba, paramecium, excrete waste via diffusion through the body surface. Freshwater protozoans use a contractile vacuole to remove excess water and maintain osmotic balance. 2.2 Platyhelminths. Flame cells. Protonephridia. Flatworms, e.g., planaria, use flame cells, protonephridia, for osmoregulation and excretion. These cells have ciliated ducts that propel waste into tubules, eventually exiting through excretory pores. 2.3 Annelids. Metanephridia. Earthworms and marine annelids have metanephridia, which are highly vascularized tubules. The nephrostome collects coulombic fluid, filters waste, and expels it through nephridia pores. 2.4 Arthropods. Malpighian tubules and green glands. Insects, g. Grasshoppers possess Malpighian tubules that extract waste from hemolymph and release uric acid into the gut for excretion with feces. Crustaceans, e.g., crabs, excrete via green glands, antennal glands, that filter waste from the hemolymph. 2.5 Mollusks. Nephridia. Kidney-like structures. Mollusks excrete via paired nephridia that filter coulombic fluid and release waste near the mantle cavity. 2.6 Echinoderms. Water vascular system and polyp. Starfish lack a dedicated excretory organ and use the water vascular system and polyp dermal gills for excretion and gas exchange. 2.7 Vertebrates. Kidneys and specialized structures. Vertebrates excrete nitrogenous waste through specialized kidneys, adapted to different excretion strategies. Freshwater fish excrete large volumes of dilute urine to prevent water influx. Marine fish excrete small amounts of concentrated urine to conserve water. Birds and reptiles excrete uric acid with minimal water loss. Mammals use highly efficient nephrons for urine formation and water conservation. 3. Human excretory system. Kidney and urine formation. 3.1 Structure of the kidney. The human kidney is a bean-shaped organ responsible for urine formation. It consists of cortex, outer region, and medulla, inner region, with nephrons as the functional units. 3.2 Urine formation. 3-step process. Glomerular filtration. Blood is filtered in the glomerulus Bowman's capsule via hydrostatic pressure. The filtrate primary urine contains water, glucose, salts, and urea. Tubular reabsorption. Essential substances: glucose, amino acids, water, or reabsorbed in the proximal convoluted tubule (PCT). Tubular secretion. Excess ions H+, K+, and H4+, and toxins are secreted into the distal convoluted tubule (DCT) and collecting ducts, forming final urine. 3.3 Hormonal regulation of kidney function. Antidiuretic hormone ADH promotes water reabsorption in the collecting ducts, reducing urine volume. Aldosterone enhances sodium reabsorption, indirectly increasing water retention. Atrial natriuretic peptide ANP opposes aldosterone, reducing sodium reabsorption and promoting urine output. Previous year question. Question: describe the major modes of excretion in animals and explain how the human kidney regulates excretion and water balance. Answer: animals excrete nitrogenous waste via ammonotalism, ureotalism, and uricotalism, depending on habitat and water availability. Ammonotelic animals, e.g., fish, excrete ammonia directly into water. Ureotelic animals, e.g., mammals, convert ammonia into urea for water conservation. Uricotelic animals, e.g., birds, excrete uric acid as an insoluble paste. The human kidney maintains water balance and excretion through filtration. Blood is filtered in the glomerulus. Reabsorption. Essential substances are reabsorbed in the tubules. Secretion. Toxins and ions are secreted into urine. Hormonal control. ADH and aldosterone regulate water retention, ensuring homeostasis. Kidney function ensures electrolyte balance, pH regulation, and toxin elimination, making it vital for survival. Multiple choice questions MCQs 1. Which of the following animals is ureotelic? A frog. B. Amoeba. C. Cockroach. D. Starfish. Answer. A frog. 2. The Malpighian tubules in insects primarily function in A. Protein synthesis. B. Excretion and osmoregulation. C. Digestion. D. Respiration. Answer. B. Excretion and osmoregulation. 3. In the human kidney, the process of selective reabsorption occurs primarily in A. Glomerulus. B. Collecting duct. C. Proximal convoluted tubule. D. Loop of Henle. Answer. C. Proximal convoluted tubule. This detailed lecture provides comprehensive coverage of the comparative physiology of excretion for your University of Calicut PhD entrance exam in zoology. Let me know the next topic. You said, kidney, ChatGPT said. Kidney, structure, function, and regulation. Introduction. The kidney is a vital excretory organ in vertebrates responsible for filtering blood, removing metabolic waste, and maintaining homeostasis. It plays a critical role in osmoregulation, electrolyte balance, acid-base regulation, and hormone production. The human kidney, a bean-shaped organ, is a functional unit of the urinary system, producing urine through glomerular filtration, tubular reabsorption, and tubular secretion. 1. Structure of the kidney. Each human kidney weighs about 150 grams and is located retroperitoneally on either side of the vertebral column. It has 3 major anatomical regions. 1.1 Cortex. The outermost region containing renal corpuscles, glomeruli, and proximal and distal convoluted tubules. It is highly vascularized, allowing efficient filtration of blood. 1.2 Medulla. The inner region containing renal pyramids, separated by renal columns. Contains loop of Henle and collecting ducts, crucial for urine concentration. 1.3 Renal pelvis. The funnel-shaped structure leading to the ureter. It collects urine from the collecting ducts before passing it to the bladder. 2. Nephron, the functional unit of the kidney. Each kidney contains about 1.2 million nephrons, responsible for urine formation. A nephron consists of 2.1 Renal corpuscle filtration unit. Glomerulus, a network of capillaries where blood filtration occurs under high pressure. Bowman's capsule, a cup-shaped structure that surrounds the glomerulus, collecting filtrate primary urine. 2.2 Renal tubules, modification of filtrate. Proximal convoluted tubule PCT reabsorbs glucose, amino acids, water, and ions from filtrate into blood. Loop of Henle creates a countercurrent multiplier system, concentrating urine. Distal convoluted tubule DCT regulates pH, sodium, and potassium balance under hormonal control. Collecting duct, final site of water reabsorption, influenced by antidiuretic hormone ADH. 3. Functions of the kidney. 3.1 Excretion of metabolic waste filters urea, uric acid, creatinine, and toxins from the blood. Eliminates drugs and hormones to prevent toxicity. 3.2 Osmoregulation. Maintains the body's water balance, adjusting urine concentration based on hydration levels. The loop of Henle plays a key role in water reabsorption. 3.3 Electrolyte balance. Regulates sodium, not potassium K, chloride, Cl, and calcium Ca² levels. Prevents electrolyte imbalances, which can affect nerve and muscle function. 3.4 Acid-base regulation. Maintains blood pH 7.35-7.45 by excreting H ions and reabsorbing bicarbonate HCO. 3.5 Blood pressure regulation. The renin-angiotensin-aldosterone system ROS controls blood pressure by regulating sodium and water retention. 3.6 Hormone production. Erythropoietin EPO stimulates red blood cell production in response to low oxygen. Calcitriol active vitamin D regulates calcium and phosphate metabolism. 4. Mechanism of urine formation. 4.1 Glomerular filtration. Blood is filtered in the glomerulus under high hydrostatic pressure. The filtrate contains water, salts, glucose, urea, and amino acids but excludes large proteins in blood cells. 4.2 Tubular reabsorption. Essential substances are reabsorbed into blood: PCT, glucose, amino acids, water, and sodium. Loop of Henle, water reabsorption descending limb and salt reabsorption ascending limb. DCT and collecting duct, final water and ion adjustments, regulated by hormones. 4.3 Tubular secretion. Active transport of H, K, NH, ammonium, and toxins into the filtrate for elimination. 5. Hormonal regulation of kidney function. 5.1 Antidiuretic hormone ADH vasopressin released by the posterior pituitary in response to low water levels. Increases water reabsorption in the collecting ducts, reducing urine volume. 5.2 Aldosterone. Secreted by the adrenal cortex in response to low sodium levels. Increases NA reabsorption and K excretion, promoting water retention. 5.3 Renin-angiotensin system. Renin from the kidney converts angiotensinogen right pointing arrow angiotensin I right pointing arrow angiotensin II, which increases blood pressure by vasoconstriction. Stimulates aldosterone release for sodium and water retention. 5.4 Atrial natriuretic peptide ANP secreted by the heart atria in response to high blood pressure. Inhibits aldosterone and ADH, promoting sodium and water excretion. 6. Clinical disorders of the kidney. 6.1 Chronic kidney disease CKD. Progressive loss of kidney function, leading to uremia and electrolyte imbalances. Causes: diabetes, hypertension, infections, nephrotoxins. 6.2 Acute kidney injury AKI. Sudden loss of renal function due to shock, dehydration, or toxins. 6.3 Kidney stones nephrolithiasis. Hard deposits of calcium oxalate, uric acid, or struvite obstructing urine flow. 6.4 Nephrotic syndrome. Excessive protein loss in urine due to glomerular damage. 6.5 Polycystic kidney disease PKD, genetic disorder causing cyst formation, impairing renal function. Previous year question. Question: describe the structure and function of the nephron and its role in urine formation. Answer: the nephron is the functional unit of the kidney, responsible for filtration, reabsorption, and secretion. Glomerulus and Bowman's capsule filters water, salts, and urea into the renal tubule. Proximal convoluted tubule PCT reabsorbs glucose, amino acids, and Na. Loop of Henle establishes a concentration gradient for water reabsorption. Distal convoluted tubule DCT regulates ion balance and pH. Collecting duct adjusts final urine concentration, regulated by ADH. The nephron maintains homeostasis by balancing water, electrolytes, and pH, ensuring proper waste elimination. Multiple choice questions MCQs 1. The primary site for glucose reabsorption in the nephron is a glomerulus. B. Loop of Henle. C. Proximal convoluted tubule. D. Collecting duct. Answer: C. Proximal convoluted tubule. 2. Which hormone promotes water reabsorption in the kidney? A. Insulin. B. ADHC, oxytocin D, glucagon. Answer, B. ADH. 3. The countercurrent multiplier system in the nephron is created by a proximal tubule. B. Loop of Henle. C. Collecting duct. D. Bowman's capsule. Answer, B. Loop of Henle. This detailed lecture provides maximum coverage for the University of Calicut PhD entrance exam in zoology. Let me know your next topic. You said, urine formation, ChatGPT said. Urine formation, mechanism and regulation. Introduction. Urine formation is a crucial physiological process in vertebrates that ensures the removal of metabolic waste, toxins, and excess ions while maintaining homeostasis of body fluids. The kidneys regulate the composition and volume of extracellular fluid through urine production. This process occurs in the nephrons, the structural and functional units of the kidneys, and involves glomerular filtration, tubular reabsorption, and tubular secretion. 1. Stages of urine formation. 1.1 Glomerular filtration. Ultrafiltration occurs in the renal corpuscle, which includes the glomerulus and network of capillaries and Bowman's capsule. Hydrostatic pressure 55 mm of mercury forces water, ions, glucose, amino acids, and metabolic waste urea, creatinine through the glomerular capillary walls into the Bowman's space, forming the glomerular filtrate. Large molecules like proteins in blood cells are not filtered due to the selective permeability of the glomerular membrane. Filtration rate. The glomerular filtration rate GFR is about 125 milliliters per minute, 180 L per day in a healthy adult. Regulation of GFR. Autoregulation adjusts afferent and efferent arterial diameter. Renin-angiotensin system increases GFR by vasoconstriction. Sympathetic nervous system reduces GFR during stress. 1.2 Tubular reabsorption. About 99% of the filtrate is reabsorbed into the blood to prevent excessive loss of essential substances. Occurs primarily in the proximal convoluted tubule PCT, but continues in the loop of Henle, distal convoluted tubule DCT, and collecting ducts. Mechanisms of reabsorption. Active transport requires energy, e.g., NA reabsorption via NA pump. Passive diffusion, movement along concentration gradients. Osmosis, water reabsorption via aquaporins, water channels. Key reabsorbed substances. PCT, 65-70% of Na, Cl, glucose, amino acids, and water. Loop of Henle, water descending limb, Na and Cl ascending limb. DCT and collecting duct, final adjustments via hormonal regulation, ADH, aldosterone. 1.3 Tubular secretion. Active transport of waste products and excess ions from blood into the renal tubule. Mainly occurs in DCT and collecting ducts. Key secreted substances, H ions, maintains blood pH balance. K ions, regulated by aldosterone. Urea and ammonia, detoxification. Drugs and toxins, penicillin, creatinine, and organic acids are actively secreted. 2. Role of hormones in urine formation. 2.1 Antidiuretic hormone, ADH, vasopressin. Secreted by the posterior pituitary in response to low blood volume or high plasma osmolarity. Increases water reabsorption by inserting aquaporins in the collecting ducts, reducing urine output. 2.2 Aldosterone. Released by the adrenal cortex in response to low sodium levels or high potassium levels. Increases NA reabsorption and K excretion, enhancing water retention. 2.3 Renin-angiotensin system. Renin from juxtaglomerular cells activates angiotensin, which constricts blood vessels, increases blood pressure. Stimulates aldosterone release, enhancing NA and water retention. 2.4 Atrial natriuretic peptide, ANP, secreted by the heart atria when blood volume increases. Inhibits NA reabsorption, leading to increased urine output, diuresis. 3. Concentration of urine, countercurrent mechanism. The loop of Henle creates a countercurrent multiplier system. The descending limb is permeable to water but not solutes. The ascending limb is impermeable to water but actively pumps Na and Cl into the medulla. This establishes a hyperesmotic gradient, allowing water reabsorption in the collecting duct under ADH influence. The vasorecta countercurrent exchanger maintains the medullary concentration gradient, preventing dilution of the renal medulla. 4. Urine composition. Water, approximately 95%. Solutes. Urea 2%, nitrogenous waste from protein metabolism. Electrolytes, Na, K, Cl, Ca squared, Mg squared, So squared, Po cubed. Creatinine, muscle metabolism waste. Uric acid, breakdown of nucleic acids. Hormones and drugs, cleared from circulation. Normal urine output, 1 to 2 L per day. Abnormalities. Glucose in urine, glycosuria, diabetes mellitus. Protein in urine, proteinuria, kidney damage. Blood in urine, hematuria, infection, kidney stones. 5. Clinical disorders related to urine formation. 5.1 Polyuria. Excessive urination, often due to diabetes mellitus or insipidus. 5.2 Oliguria and anuria. Oliguria, low urine output and LT, 400 milliliters per day. Anuria, complete cessation of urine formation. 5.3 Nephritis. Inflammation of the kidney, e.g., glomerulonephritis. 5.4 Renal failure. Acute, sudden loss of kidney function. Chronic, progressive kidney damage requiring dialysis. 5.5 Kidney stones, renal calculi formed from calcium oxalate, uric acid, or struvite. Causes pain and urine obstruction. Previous year question solved. Question, explain the role of the nephron in the formation of urine and the hormonal regulation involved. Answer. Urine formation occurs in the nephrons and consists of 3 main processes. Glomerular filtration. Blood is filtered in the glomerulus under high pressure, producing glomerular filtrate. Tubular reabsorption. Essential nutrients, glucose, amino acids, Na, and water are reabsorbed in the PCT, loop of Henle, and DCT. Tubular secretion. Unwanted substances H, K, urea are actively secreted into the DCT and collecting duct. Hormonal regulation. ADH promotes water reabsorption in the collecting duct. Aldosterone enhances NA reabsorption and K excretion. Renin-angiotensin system increases GFR and blood pressure. ANP reduces NA and water retention. The countercurrent mechanism in the loop of Henle ensures urine concentration, maintaining fluid and electrolyte balance. Multiple choice questions, MCQs. 1. The hormone responsible for reducing urine output during dehydration is A. Oxytocin. B. ADH. C. Aldosterone. D. ANP. Answer, B. ADH. 2. The site of maximum water reabsorption in the nephron is A. Loop of Henle. B. Proximal convoluted tubule. C. Distal convoluted tubule. D. Collecting duct. Answer, B. Proximal convoluted tubule. 3. The countercurrent multiplier is primarily established in A. Glomerulus. B. Proximal tubule. C. Loop of Henle. D. Collecting duct. Answer, C. Loop of Henle. This detailed lecture ensures maximum coverage for your PhD entrance exam in zoology. Let me know the next topic. You said, urine. Concentration, waste elimination, micturition, regulation of water balance, ChatGPT said. Urine concentration, waste elimination, micturition, and regulation of water balance 1. Urine concentration, the countercurrent mechanism. Urine concentration is a vital process that helps the body conserve water while ensuring the removal of metabolic waste. This process primarily occurs in the loop of Henle and collecting ducts, which function through the countercurrent multiplier and exchanger system. 1.1 Countercurrent multiplier, loop of Henle. The descending limb is permeable to water but impermeable to solutes. Water moves out by osmosis, concentrating the filtrate. The ascending limb is impermeable to water but actively transports non-Cl into the medullary interstitium, creating a hyperosmotic gradient. This establishes a medullary concentration gradient, essential for water reabsorption in later stages. 1.2 Countercurrent exchanger, vasa recta. The vasa recta, a network of capillaries surrounding the loop of Henle, maintains the concentration gradient by removing excess water and solutes without disrupting the osmotic balance. This mechanism ensures efficient reabsorption of water when needed, leading to concentrated urine production under the influence of antidiuretic hormone ADH. 1.3 Role of antidiuretic hormone ADH. ADH vasopressin is secreted by the posterior pituitary when blood osmolarity is high dehydration. It increases the permeability of the collecting duct by inserting aquaporins, water channels, allowing water reabsorption. This results in the production of concentrated urine. In the absence of ADH, water is not reabsorbed, and dilute urine is excreted. 2. Waste elimination. The urinary system plays a critical role in excreting metabolic wastes that result from cellular metabolism. 2.1 Major waste products in urine: urea. Derived from protein metabolism in the liver, urea cycle. Eliminates excess nitrogen. Creatinine. Product of muscle metabolism, creatine phosphate breakdown. High levels indicate kidney dysfunction. Uric acid. Produced from purine DNA, RNA metabolism. High levels cause gout, uric acid crystal deposition in joints. Ions. Excess Na, K, Ca squared, Cl, and HCl help maintain electrolyte balance. Drugs and toxins. Antibiotics, metabolites, and synthetic compounds are filtered out. Importance of waste elimination. Prevents toxic buildup in the body. Maintains acid-base and electrolyte balance. Regulates osmotic pressure and prevents cell damage. 3. Micturition, the process of urination. Micturition is the process of emptying the urinary bladder. It involves voluntary and involuntary control through the nervous system. 3.1 Anatomy of the bladder and urethrourinary bladder. Stores urine, approximately 500 milliliters capacity in adults. Detrusor muscle, smooth muscle layer that contracts during urination. Internal urethral sphincter, involuntary control, smooth muscle. External urethral sphincter, voluntary control, skeletal muscle. 3.2 Steps of micturition. Filling phase. Urine collects in the bladder, causing stretch receptors to send signals to the spinal cord. Storage phase. The sympathetic nervous system, SNS, keeps the internal sphincter contracted and relaxes the detrusor muscle, preventing urination. Micturition reflex, voiding phase. When the bladder reaches approximately 300 to 400 ml, parasympathetic nerves stimulate detrusor contraction and internal sphincter relaxation. The external sphincter, voluntary, is consciously relaxed to allow urination. Post-micturition phase. The bladder returns to its relaxed state, and the sphincters close. 3.3 Neural control of micturition, spinal reflex, S2S4, coordinates involuntary bladder control. Pontine micturition center, PMC, coordinates voluntary control. Cerebral cortex, allows conscious decision-making about urination. 3.4 Disorders of micturition. Urinary incontinence, loss of bladder control. Urinary retention, inability to empty the bladder. Neurogenic bladder, damage to nerves controlling micturition, e.g., spinal cord injury. 4. Regulation of water balance. The kidneys play a key role in maintaining fluid homeostasis through hormonal control and osmoregulation. 4.1 Hormonal regulation of water balance. Antidiuretic hormone, ADH vasopressin. Increases water reabsorption in the collecting ducts. Released when blood osmolarity increases. Dehydration. Deficiency leads to diabetes insipidus, excessive urination and dehydration. Aldosterone. Secreted by the adrenal cortex. Enhances Na reabsorption. Water follows osmotically, reducing urine output. Regulated by the renin-angiotensin system, ROS. Atrial natriuretic peptide, ANP. Secreted by the heart atria when blood volume is high. Inhibits Na reabsorption, leading to increased urine output. Diuresis. Renin-angiotensin system, RAAS. Renin from kidneys converts angiotensinogen to angiotensin I, which is converted to angiotensin II. Angiotensin II increases blood pressure and stimulates aldosterone secretion. Previous year question, solved. Question, explain the role of the countercurrent mechanism in urine concentration and water balance. Answer. The countercurrent mechanism, present in the loop of Henle, is essential for urine concentration and water conservation. Countercurrent multiplier, loop of Henle. The descending limb is permeable to water, causing water to leave the tubule due to the hyperosmotic medulla. The ascending limb actively transports non-Cl out, creating a medullary concentration gradient. Countercurrent exchanger, vasa recta. The vasa recta preserves this gradient by removing excess solutes in water without disrupting osmolarity. ADH and urine concentration. ADH enhances water permeability in the collecting duct, allowing more water reabsorption. This leads to hyperosmotic concentrated urine during dehydration and hyposmotic dilute urine when water intake is high. This mechanism ensures water balance in the body and prevents dehydration. Multiple choice questions, MCQs. 1. Which hormone increases water reabsorption in the collecting duct? A. Aldosterone. B. ADH. C. ANPD. Renin. Answer. B. ADH. 2. The main nitrogenous waste excreted in human urine is ammonia. B. Uric acid. C. Urea. D. Creatinine. Answer. C. Urea. 3. Which structure in the nephron plays the most important role in urine concentration? A. Proximal tubule. B. Distal tubule. C. Loop of Henle. D. Glomerulus. Answer. C. Loop of Henle. This detailed lecture ensures maximum topic coverage for your PhD entrance exam in zoology. Let me know the next topic. You said, blood volume. Blood pressure, electrolyte balance, acid-base balance. ChatGPT said. Blood volume, blood pressure, electrolyte balance, and acid-base balance 1. Blood volume, regulation and importance 1.1 definition and normal range. Blood volume refers to the total amount of blood, plasma plus cells circulating in the body. In an average adult, it is approximately 5 to 6 liters, 7-8% of body weight. 1.2 Factors regulating blood volume. Blood volume is regulated by renal function, hormones, and fluid intake output. Renal regulation, kidney function. Glomerular filtration rate, GFR, controls how much plasma is filtered. Renin-angiotensin-aldosterone system, RAS, increases non-water retention. Antidiuretic hormone, ADH, promotes water reabsorption in the collecting ducts. Hormonal control. Aldosterone, increases NA retention, leading to water retention. ADH, vasopressin, reduces urine output by increasing water absorption. Atrial natriuretic peptide, ANP, reduces blood volume by increasing non-water excretion. Capillary exchange in plasma proteins. Albumin, maintains oncotic pressure, preventing excess fluid loss into tissues. Hydration and fluid intake. Excess fluid increases blood volume, whereas dehydration reduces it. 1.3 Effects of abnormal blood volume. Hypovolemia, low blood volume, causes dehydration, shock, and low blood pressure. Hypervolemia, high blood volume, leads to hypertension, edema, and congestive heart failure. 2. Blood pressure, mechanism and regulation. 2.1 Definition and normal range. Blood pressure, BP, is the force exerted by circulating blood on arterial walls. Normal BP is 120/80ths of a millimeter of mercury in adults. Systolic pressure, 120 millimeters of mercury, maximum pressure during heart contraction. Diastolic pressure, 80 millimeters of mercury, minimum pressure during relaxation. 2.2 Factors affecting blood pressure, cardiac output, CO, BP increases if the heart pumps more blood. Peripheral resistance, narrower arteries increase BP. Blood volume, higher volume leads to higher BP. Blood viscosity, thicker blood, high RBCs, increases resistance in BP. 2.3 Neural and chemical regulation of blood pressure, neural control. Baroreceptors in carotid artery and aorta detect BP changes and adjust heart rate and vessel dilation. Sympathetic nervous system, SNS, increases heart rate and vasoconstriction, raises BP. Parasympathetic nervous system, PNS, reduces heart rate, lowers BP. Hormonal regulation. RAS, renin-angiotensin-aldosterone system, increases BP via vasoconstriction and NA retention. ADH, retains water, increasing BP. ANP, promotes NA excretion, lowering BP. 2.4 Hypertension versus hypotension. Hypertension, high BP, leads to heart disease, stroke, kidney failure. Hypotension, low BP, causes dizziness, fainting, and shock. 3. Electrolyte balance, role in regulation. 3.1 Importance of electrolytes. Electrolytes are ions that help in nerve transmission, muscle contraction, fluid balance, and pH regulation. 3.2 Key electrolytes and their function. Sodium, Na. Major extracellular ion, regulated by aldosterone, maintains blood volume and BP. Potassium, K, major intracellular ion, essential for muscle contraction, heart rhythm, regulated by kidneys. Calcium, Ca squared, needed for muscle contraction, nerve function, and clotting, controlled by parathyroid hormone, PTH. Chloride, Cl, helps maintain acid-base balance. Bicarbonate, HCO, major buffer in acid-base balance. 3.3 Electrolyte imbalances. Hyponatremia, low Na, causes confusion, weakness. Hyperkalemia, high K, leads to cardiac arrhythmia. Hypocalcemia, low Ca squared, causes muscle spasms. 4. Acid-base balance, pH homeostasis. 4.1 Normal pH and importance. The body maintains a blood pH of 7.35-7.45. Any deviation can disrupt enzyme activity, oxygen transport, and nerve function. 4.2 Buffer systems in the body. Bicarbonate buffer, HCO, primary buffer in the blood. Maintains pH by converting H into carbonic acid, HCO, which dissociates into CO and HO. HCO plus H, right pointing arrow, HCO, right pointing arrow, CO plus HO. Protein buffer. Hemoglobin and plasma proteins bind H to regulate pH. Phosphate buffer. Important in intracellular fluid and kidney regulation. 4.3 Role of lungs and kidneys in pH regulation. Lungs control CO removal. Faster breathing raises pH. Kidneys excrete H and reabsorb HCO to maintain balance. 4.4 Acidosis versus alkalosis. Acidosis, low pH and LT, 7.35, caused by respiratory failure, CO retention, or kidney dysfunction, H retention. Alkalosis, high pH and GT, 7.45, caused by hyperventilation, excess CO loss, or excess HCO. Previous year question solved. Question: describe the role of the kidney in blood volume and acid-base balance. Answer: the kidney plays a crucial role in regulating blood volume and maintaining acid-base balance through filtration, reabsorption, and hormonal control. Blood volume regulation. The RAS system increases non-water retention, raising blood volume. ADH promotes water reabsorption to prevent dehydration. ANP enhances non-water excretion, reducing blood volume. Acid-base balance. The kidney secretes H and reabsorbs HCO to regulate pH. Ammonia buffering in urine prevents acidosis. Respiratory compensation, CO removal, supports pH stability. Thus, kidneys ensure fluid homeostasis and prevent pH imbalances that could disrupt physiological functions. Multiple choice questions. MCQs. 1. Which hormone decreases blood volume by promoting sodium excretion? A. ADH. B. Aldosterone. C and B. D. Renin. Answer: C and B. 2. The major buffer system in the blood is a. Phosphate buffer. B. Protein buffer. C. Bicarbonate buffer. D. Ammonia buffer. Answer: C. Bicarbonate buffer. 3. The normal pH range of blood is a. 6.8 7.2. B. 7.35 7.45. C. 7.58.0. D. 6.0 6.8. Answer: B. 7.35 7.45. This detailed lecture covers all key concepts for your PhD entrance exam in zoology. Let me know the next topic. You said adaptation, comfort zone, body temperature, physical, chemical, neural regulation, acclimatization, stress and adaptation. ChatGPT said adaptation and regulation of body temperature. 1. Adaptation, concept and comfort zone. 1.1 Definition of adaptation. Adaptation is the physiological, behavioral, or morphological changes that enhance an organism's ability to survive and reproduce in a particular environment. It occurs over evolutionary time through natural selection, ensuring that organisms can function efficiently in changing environmental conditions. 1.2 Comfort zone and thermal neutral zone. The comfort zone is the range of environmental conditions in which an organism maintains homeostasis with minimal energy expenditure. In endotherms, warm-blooded animals, this corresponds to the thermal neutral zone, TNZ, the temperature range where metabolic heat production and loss are balanced without stress. For humans, the comfort zone is typically 20–25 degrees Celsius, while for other species, it varies based on evolutionary adaptation. Outside this range, organisms employ physiological and behavioral mechanisms to maintain internal stability. 2. Body temperature regulation, physical, chemical, and neural mechanisms. 2.1 Types of thermoregulation. Endotherms, homeotherms maintain a constant body temperature through metabolic heat production, e.g., mammals, birds. Ectotherms, poikilotherms body temperature fluctuates with the environment, e.g., reptiles, amphibians. Heterotherms exhibit characteristics of both groups, such as hibernators, e.g., bats, some rodents. 2.2 Physical regulation of body temperature. Physical mechanisms involve external adjustments to control heat exchange. Insulation. Fur, feathers, and fat blubber reduce heat loss in cold environments. Desert animals have thin coats to promote heat dissipation. Evaporative cooling. Sweating, used by humans and some animals to release heat. Panting, common in dogs and birds to cool through rapid evaporation. Behavioral adjustments. Seeking shade, burrowing, or changing posture to regulate heat exchange. Basking in the sun, cold-blooded animals, or hibernation bears to adapt to cold. 2.3 Chemical regulation of body temperature. Metabolic processes play a major role in temperature homeostasis. Basal metabolic rate, BMR, determines the minimum energy required for vital functions, higher in endotherms, allowing them to maintain constant body temperature. Thermogenesis. Shivering thermogenesis, muscle contractions generate heat. Non-shivering thermogenesis, brown adipose tissue fat burns fat to produce heat, especially in newborns and hibernators. Hormonal control. Thyroxine T and triiodothyronine T increase metabolic heat production. Adrenaline and noradrenaline activate thermogenic responses in cold stress. 2.4 Neural regulation of body temperature. The hypothalamus acts as the body's thermoregulatory center, maintaining homeostasis through autonomic and behavioral responses. Heat loss mechanisms, high temperature. Vasodilation, blood vessels widen to release heat. Sweating, evaporation of sweat cools the body. Reduced metabolism, limits heat production. Heat retention mechanisms, cold temperature. Vasoconstriction, blood vessels constrict to reduce heat loss. Shivering, generates heat through muscle activity. Piloerection, goosebumps, traps warm air near the skin. Hypothalamic temperature sensors. Detect internal temperature changes and trigger corrective responses via the autonomic nervous system, ANS. 3. Acclimatization, short-term adaptation. Acclimatization is the physiological adjustment to changing environmental conditions, often occurring over days to weeks. 3.1 Acclimatization to cold. Increased metabolic rate, enhanced thyroxine and adrenaline secretion. Enhanced vasoconstriction to reduce heat loss. Increased insulation via fur growth or fat deposition. 3.2 Acclimatization to heat. Increased sweating efficiency, faster onset, more dilute sweat. Reduced electrolyte loss, better NOx conservation. Enhanced cardiovascular efficiency, better heat dissipation. 3.3 High altitude acclimatization. At high altitudes above 2,500 meters, oxygen levels are lower, triggering adaptations. Increased red blood cell RBC production, erythropoietin stimulation. Enhanced lung ventilation, faster breathing. Higher capillary density for better oxygen delivery. 4. Stress and adaptation. 4.1 Definition of stress. Stress is any external or internal challenge that disrupts homeostasis. It can be physical temperature, injury, toxins, physiological, hormonal imbalance, or psychological fear, anxiety. 4.2 General adaptation syndrome, GAS, proposed by Hans Selye. The three-stage response to stress includes alarm phase, activation of the sympathetic nervous system, SNS, and hypothalamic pituitary adrenal, HPA, axis, release of adrenaline and cortisol to prepare for fight or flight. Resistance phase. Body adapts to stress through increased energy production and homeostasis maintenance. Exhaustion phase. Prolonged stress leads to immune suppression, fatigue, and disease. Previous year question solved. Question. Explain the role of the hypothalamus in body temperature regulation. Answer. The hypothalamus acts as the primary thermoregulatory center, integrating information from internal core temperature and external skin receptors sources to maintain homeostasis. Heat loss mechanisms when body is hot. Vasodilation expands blood vessels to release heat. Sweating, evaporation cools the skin. Reduced metabolic rate minimizes internal heat production. Heat retention mechanisms when body is cold. Vasoconstriction narrows blood vessels to retain heat. Shivering, muscle contraction, increasing metabolic heat production. Insulin. B. Thyroxine. C. Oxytocin. D. Vasopressin. Answer. B. Thyroxine. 2. Thermal neutral zone, TNZ, refers to the range of environmental temperatures where the metabolic rate remains constant. B. Sweating occurs excessively. C. Shivering is activated. D. The body loses heat rapidly. Answer. The metabolic rate remains constant. 3. Which physiological mechanism is used by desert animals to avoid overheating? Vasoconstriction. B. Non-shivering thermogenesis. C. Evaporative cooling. D. Brown fat metabolism. Answer. C. Evaporative cooling. This detailed lecture covers adaptation, body temperature regulation, acclimatization, and stress response comprehensively for your PhD entrance exam in zoology. Let me know the next topic for an in-depth study. You said. Endocrinology, endocrine glands. ChatGPT set. 1. Adaptation, concept and comfort zone. 1.1 Definition of adaptation. Adaptation is the physiological, behavioral, or morphological changes that enhance an organism's ability to survive and reproduce in a particular environment. It occurs over evolutionary time through natural selection, ensuring that organisms can function efficiently in changing environmental conditions. 1.2 Comfort zone and thermal neutral zone. The comfort zone is the range of environmental conditions in which an organism maintains homeostasis with minimal energy expenditure. In endotherms, warm-blooded animals, this corresponds to the thermal neutral zone, TNZ, the temperature range where metabolic heat production and loss are balanced without stress. For humans, the comfort zone is typically 20–25 degrees Celsius, while for other species, it varies based on evolutionary adaptation. Outside this range, organisms employ physiological and behavioral mechanisms to maintain internal stability. 2. Body temperature regulation, physical, chemical, and neural mechanisms. 2.1 Types of thermoregulation. Endotherms, homeotherms maintain a constant body temperature through metabolic heat production, e.g., mammals, birds. Ectotherms, poikilotherms body temperature fluctuates with the environment, e.g., reptiles, amphibians. Heterotherms exhibit characteristics of both groups, such as hibernators, e.g., bats, some rodents. 2.2 Physical regulation of body temperature. Physical mechanisms involve external adjustments to control heat exchange. Insulation. Fur, feathers, and fat blubber reduce heat loss in cold environments. Desert animals have thin coats to promote heat dissipation. Evaporative cooling. Sweating, used by humans and some animals to release heat. Panting, common in dogs and birds to cool through rapid evaporation. Behavioral adjustments. Seeking shade, burrowing, or changing posture to regulate heat exchange. Basking in the sun, cold-blooded animals, or hibernation bears to adapt to cold. 2.3 Chemical regulation of body temperature. Metabolic processes play a major role in temperature homeostasis. Basal metabolic rate, BMR. Determines the minimum energy required for vital functions. Higher in endotherms, allowing them to maintain constant body temperature. Thermogenesis. Shivering thermogenesis. Muscle contractions generate heat. Non-shivering thermogenesis. Brown adipose tissue fat burns fat to produce heat, especially in newborns and hibernators. Hormonal control. Thyroxine T and triiodothyronine T increase metabolic heat production. Adrenaline and noradrenaline activate thermogenic responses in cold stress. 2.4 Neural regulation of body temperature. The hypothalamus acts as the body's thermoregulatory center, maintaining homeostasis through autonomic and behavioral responses. Heat loss mechanisms. High temperature. Vasodilation. Blood vessels widen to release heat. Sweating. Evaporation of sweat cools the body. Reduced metabolism. Limits heat production. Heat retention mechanisms. Cold temperature. Vasoconstriction. Blood vessels constrict to reduce heat loss. Shivering. Generates heat through muscle activity. Piloerection. Goosebumps. Traps warm air near the skin. Hypothalamic temperature sensors. Detect internal temperature changes and trigger corrective responses via the autonomic nervous system, ANS. 3. Acclimatization. Short-term adaptation. Acclimatization is the physiological adjustment to changing environmental conditions, often occurring over days to weeks. 3.1 Acclimatization to cold. Increased metabolic rate, enhanced thyroxine and adrenaline secretion, enhanced vasoconstriction to reduce heat loss. Increased insulation via fur growth or fat deposition. 3.2 Acclimatization to heat. Increased sweating efficiency, faster onset, more dilute sweat. Reduced electrolyte loss, better NOx conservation. Enhanced cardiovascular efficiency, better heat dissipation. 3.3 High altitude acclimatization. At high altitudes, above 2,500 meters, oxygen levels are lower, triggering adaptations. Increased red blood cell, RBC, production, erythropoietin stimulation. Enhanced lung ventilation, faster breathing. Higher capillary density for better oxygen delivery. 4. Stress and adaptation. 4.1 Definition of stress. Stress is any external or internal challenge that disrupts homeostasis. It can be physical temperature, injury, toxins, physiological, hormonal imbalance, or psychological fear, anxiety. 4.2 General adaptation syndrome, GAS, proposed by Hans Selye. The three-stage response to stress includes alarm phase. Activation of the sympathetic nervous system, SNS, and hypothalamic pituitary adrenal, HPA, axis. Release of adrenaline and cortisol to prepare for fight or flight. Resistance phase. Body adapts to stress through increased energy production and homeostasis maintenance. Exhaustion phase. Prolonged stress leads to immune suppression, fatigue, and disease. Previous year question solved. Question. Explain the role of the hypothalamus in body temperature regulation. Answer. The hypothalamus acts as the primary thermoregulatory center, integrating information from internal core temperature and external skin receptors sources to maintain homeostasis. Heat loss mechanisms when body is hot. Vasodilation expands blood vessels to release heat. Sweating, evaporation cools the skin. Reduced metabolic rate minimizes internal heat production. Heat retention mechanisms when body is cold. Vasoconstriction narrows blood vessels to retain heat. Shivering, muscle contractions generate warmth. Piloerection, goosebumps, traps warm air for insulation. The hypothalamus ensures body temperature stability by regulating autonomic, endocrine, and behavioral responses to environmental changes. Multiple choice questions, MCQs. 1. Which of the following hormones is primarily responsible for increasing metabolic heat production? A. Insulin. B. Thyroxine. C. Oxytocin. D. Vasopressin. Answer. B. Thyroxine. 2. The thermal neutral zone, TNZ, refers to the range of environmental temperatures where the metabolic rate remains constant. B. Sweating occurs excessively. C. Shivering is activated. D. The body loses heat rapidly. Answer. The metabolic rate remains constant. 3. Which physiological mechanism is used by desert animals to avoid overheating? A. Vasoconstriction. B. Non-shivering thermogenesis. C. Evaporative cooling. D. Brown fat metabolism. Answer. C. Evaporative cooling. This detailed lecture covers adaptation, body temperature regulation, acclimatization, and stress response comprehensively for your PhD entrance exam in zoology. Let me know the next topic for an in-depth study. You said. Endocrinology, endocrine glands. ChatGPT said. Endocrinology, endocrine glands 1. Introduction to endocrinology. Endocrinology is the branch of biology that studies the structure, function, and regulation of endocrine glands and hormones. Unlike exocrine glands, which release secretions through ducts, endocrine glands are ductless and release hormones directly into the bloodstream to regulate physiological processes such as metabolism, growth, reproduction, and homeostasis. Hormones act as chemical messengers that bind to specific target cells via receptors, triggering a cascade of biological responses. The endocrine system works in coordination with the nervous system to maintain internal balance and respond to external stimuli. 2. Major endocrine glands and their functions. 2.1 Hypothalamus, the master regulator. The hypothalamus is a neuroendocrine organ located in the brain that connects the nervous and endocrine systems. It regulates hormone secretion from the pituitary gland through releasing and inhibiting hormones. Thyrotropin-releasing hormone, TRH, right-pointing arrow stimulates TSH secretion from the pituitary. Gonadotropin-releasing hormone, GnRH, right-pointing arrow stimulates LH and FSH release. Corticotropin-releasing hormone, CRH, right-pointing arrow stimulates ACTH secretion. Growth hormone-releasing hormone, GHRH, right-pointing arrow stimulates GH release. Somatostatin, GHIH, right-pointing arrow inhibits GH and TSH release. 2.2 Pituitary gland, the master gland. The pituitary gland is a pea-sized gland located at the base of the brain and divided into two parts. A. Anterior pituitary adenohypophysis, it secretes tropic hormones that regulate other endocrine glands. Growth hormone, GH, promotes growth, metabolism, and protein synthesis. Thyroid-stimulating hormone, TSH, stimulates the thyroid gland to produce thyroxine. Adrenocorticotropic hormone, ACTH, stimulates the adrenal cortex to produce cortisol. Follicle-stimulating hormone, FSH, regulates gametogenesis in gonads. Luteinizing hormone, LH, stimulates ovulation and testosterone secretion. Prolactin, PRL, stimulates milk production in mammary glands. B. Posterior pituitary, neurohypophysis. The posterior pituitary does not synthesize hormones but stores and releases. Oxytocin,